The next talk will be delivered by Professor Ajit Kumar Shasany. I invite you, sir. And Dr. Ajit Kumar Shasany is currently the director of CSR NBRL Lucknow, and he previously served as director of ICR NIPB New Delhi and also chief scientist at CMAP. His credit goes, he had done pioneering work on the genome sequencing of Ossimum sanctum, the holy basil plant of India. To his credit, he has got 146 publications and 101 patents, and also he has led 30 research projects. His area of research is on aromatic plant technologies, climate resilient crops, and plant immunity. Uh, Dr. Shashni is also a fellow of National Academy of Agriculture Science and also National Academy of Sciences, along with a lot of technology prizes. So I welcome you, sir. Thank you.
First of all, I thank Ajit and Madam Soumya for whom I am here. I have not seen this institute, but speaking about this mathematical science, but working on biology and doing wonderful work. And I knew first when they produced that impact software of medicine aromatic plant. And Madam Usha is here, Subra Madam, Sodamini, Dada, and all other people, students and friends. It's a privilege to speak to you. But what I'll do, I'll give you a concept. I'll not speak much about the genome or genomics as such, but in between it will come automatically. So the concept is internal immunity. I am not talking about innate immunity. I am talking about the internal immunity and how secondary metabolites they help. But before that, I'll speak something about my institute. So this is the institute which in 78 it became National Botanical Garden. It came under CSIR, but it's an old institute, 200 old year botanical garden by the Nawabs of Awadh. It has transferred hands up Awadh, Nawab Awadh, then that England government of UK, then came to government of India, then came to CSIR after all. So we work on lesser known plants. The plants not limited to crop plants also. We work on crop plants but to a limited extent. But we work on the potential plants which are not being seen the daylight, indigenous plants, we have to bring to the fore and make them utilizable. So we have departments like taxonomy, we identify, somebody was asking ecological agriculture. So ecology, because this is a unique institute of National Botanical Research Institute. It is different from Botanical Survey of India. They do survey, we do research. So starting from plant taxonomy, plant ecology, behavior of the plants, microbes and soil, what you are, aptly the title is. Then coming to the biodiversity, how biodiversity is shrinking, how biodiversity is increasing, how we can utilize the biodiversity, and what is the ecology for this biodiversity. We always think of a plant which is going in a specific area. That means the speciation occurred. The speciation occurred because the plants tried to interact with the particular environment and grew accordingly. So that's all the botanists they believe. Though I am from agriculture, I am also an agricultural botanist. So coming to the other area where we work is environment. In environment, we work on microplastics, how they are actually harming us, how it is entering into the body, and how we will ameliorate the effect of microplastic in the soil, how you can take them out. Also the environment which plants actually having high positive potential, so that to attract the particulate matter of negative potential, to reduce the pollution in the air and providing higher amount of oxygen. So those type of research we do. Then coming back to again the research microbiology, environment, when you talk about the environment, Without microbes, there is no environment. We grow microorganisms within our body, and also the plants, they have microorganisms in their body. So we work on PGPRs, we work on endophytes, how they are actually helping the plants. Similarly, we have 5 bacteria, because we talk about organic farming always. So we have biofertilizers, improved biofertilizers. These improved biofertilizers, not only you get the biofertilizer in the lab, but it has to be utilizable. So these biofertilizers are rhizobium, improved strains, azospirillum, improved strain, phosphate solubilizer, and potash mineralizer. And you will not believe it, you Uttar Pradesh government, we have transferred this technology, and every district headquarter, the training is being given, the cultures are being given, the agriculture department, they produce the biofertilizer. Last 2 years, we have given 37 lakhs packet of biofertilizer, 200 gram each. That means it amounts to around 12 lakh acres of land, organic farming. Seed treatment, when you do the seed treatment with the fertilizer, they go automatically to the seed. In addition to that, we have developed 5 bacteria which are against pathogenic fungi. So when you talk about organic cultivations and agriculture in future, you have to work on that. So you cannot go for pesticides for organic cultivation. So you require biological mechanism, biostimulant, as well as the biofertilizer, as well as the organism which can control the insect pest and bacteria and pathogenic bacteria and fungi. So in this direction, having said so, we have floriculture mission. We work all over India. Even if in Tamil Nadu, we have developed a garden in Rameswaram temple. So we believe in the temple economy, small scale temple economy. Within and around the temple, if you have clusters of people, cluster of land, and the people, if you help them, give the plants, again the ecological system, thinking about the local plants to grow, and the local plants which can be utilizable, you empower the people around the temple, and whatever flowers coming out of the temple, you should have utilization mechanism, like preparing perfume oil, by preparing liquid sindoor, by preparing many other materials, dyes out of the flowers, which can be, you can dye your clothes. So these are the things you empower. So it will be a circular utilization of everything. Then conservation of threatened plants, underutilized plants for bioprospection, climate resilient plants for indoor pollution, arsenic risk assessment. Arsenic, just today morning I was talking, West Bengal is infested with arsenic, mostly for rice. So with the help of our institute, a variety has been developed, and West Bengal government and our institute, agriculture department, Muktasri, that variety is now cultivated in West Bengal, which accumulates less lysine. Not only that, our scientists, they are working on different mechanism of arsenic remediation from the soil by methylating bacteria, which can methylate it, and when you give higher amount of higher stress, that means heat, then they will evaporate to the air. In addition to that, there are some plants, weed plants, which accumulates, which take away the arsenic from the soil, because of some of the receptors are different. Arsenic receptors and transporters are different, and I am not going to talk about, but I am just giving a glimpse what work is going on. Simultaneously, we are working on genome editing of different plants like tomato for stress resistance, stress tolerance, synchronous flowering of cotton, and many more things we are working on genome editing. So this is under genetic improvement of plant resources. CSR floriculture mission, I told you, we work on CSR floriculture mission, and if you see the productivity, it has gone up a large fold, a huge productivity increase is there since the time of independence and today. Thanks to Professor Swaminathan, for the green revolution. But our area remains same. Productivity has gone up. Productivity has gone up and production has gone up in cereals, in carbohydrate crops. And as Madam was telling today morning, once we are increasing the production of productivity, the metal, the required metal, iron and zinc, which are required, they are gradually decreasing in the grain, what we are eating. And you remember your childhood days, or you remember now your parents or grandparents, they might be telling you that earlier food was tasting better. Now you are eating from Swiggy. How it tastes, I don't know. So taste not only depends upon the mineral, amino acids, this also depends upon the secondary metabolites. And nowadays, I may say that we are losing our immunity. During the COVID period, we were using all kadhaj without knowing what it does. How many people are using kadhaj, drinking kadhaj? Can you raise your hand? So these molecules are from plants, and we are intelligent people. We are using these molecules for our benefit to increase our own immunity. Why not to utilize those molecules for the plant immunity, for organic farming and natural farming in future? And what are the secondary metabolites? That concept I want to give you today. So climate change, everybody says, all the seminar, symposium, you say, go, you will listen about the climate change. Also, Article 47 says that all the citizens, you have to provide the nutritious food. One concept is synteny. Synteny means in breeding, if two genes are linked together, they are inherited generation after generation in classical synteny concept is. But the concept of synteny, I am talking about the metabolic synteny. The molecules present in one plant will be present in many other plants. If the molecule present in one plant present in many other plants, then definitely the pathway present in one plant for that molecule will be available in many other plants. So this is pathway synteny and molecular synteny. So what I have done, I have taken the help of, I have analyzed 1,000 plants. In 1,000 plants, if you see the molecules, this particular molecule is present in 550 plants. This is present in 377 plants. Like this, so the molecules are synteneous. It is not that the molecules are not there. It is the concentration of the molecule. Similarly, this is the phenyl propionate pathway. No need to enter into the pathway. You can ignore the pathway. Just listen to me, because it will create problem if you go into the pathway. So there are this is in the rosemary acid 57 plants, stilbene 10 plants, catechin 77 plants. Like this, you can find out these molecules are present in many plants. So the red line, the upstairs says these are the primary metabolites, and these are all secondary metabolites. And the plants, as I told, the compounds, suppose monoterpenes, sesquiterpenes, diterpenes, all these, if you say monoterpene producing plants, these produces monoterpene. That doesn't mean that they don't produce sesquiterpene, they produce sesquiterpene, they produce diterpenes, but they are very low amount. But the pathway is dominant. Who is going to answer? Why they are producing more monoterpene, and which ecological niche they are growing, and what interaction it is happening they are doing along with the environment. So in CIMAP, when I was a scientist, at that time Madam was there, the RC chairman also. So there was a locust infestation. Locust ate away all the crop plants which are being intercropped, but they didn't touch the medicinal plant. That's citronella, lemongrass, artemisia. All these plants, they are staying. Why? Because of the secondary metabolites. Either they don't like it, they ate something and they run away, or if they eat, they may be killed. So there is something happening with the secondary metabolites. So in the plants, so these are the armory of the plants. And you know, if I ask you a question, what is the difference between plant and animal in single sentence? We will run away when there is heat outside. We will stay inside the AC room. When it is raining, we will run away. Plant cannot move, they stay. We have an immune system starting from IgM to IgG. So if something enters into our body, antigen. But plant, they don't have that type of immune system. Hence, so they have the armory, they have developed this armory. So there are examples, insecticidal activities. So there are compounds which are having insecticidal activities. So there are compounds which are antimicrobial activity. So there are compounds already being defined. But these compounds, if you extract and spray in the field, are they stable? They may not be. For that reason, we cannot take it out and spray them. The sesquiterpene and monoterpene, if you spray them, they will evaporate. So there are problems associated with it. So why not to increase the internally this particular molecule be they are inside the plants. When insect or any other things comes, they will be inhibited. So that's the main concept behind this. So there is one example again. If you increase the hexanal, then it increases the teloge formation, and tomato, it inhibits the tomato yellow leaf curl virus. So there are examples. So after studying some of the examples, so you worked on this. Then here we have this one isoprene and alpha beta pinene and beta caryopyllene. Beta caryopyllene through jasmonic pathway, it gives immunity, and isoprene and alpha beta pinene through salicylic acid pathway, it gives immunity to the plant, to Pseudomonas. Then again, here we are actually we are collaborators, Dinesh Nagagoda and myself. We work together. We are both in Purdue University, and we work together and publish together also sometimes. So here in Ashwagandha, if we increase the expression of SIP genes, some of the SIP genes, then it will increase the udanolites, some of the udanolites, specific udanolites, and it will give tolerance. I will not tell resistance. Resistance means complete resistance. Tolerance against Pseudomonas. Similarly, farnesyl, a small compound, if you increase the expression of farnesyl in potato, it will give tolerance to late blight disease. Otherwise, it is very difficult to control these diseases. Again, I was in NIPB, National Institute of Plant Biotechnology, the same thinking went on. So we thought why the Cajanus cajan, that means the arhar, why it is susceptible to pod borer. Pod borer comes and eat it. People are working on a BT protein, expression of BT protein, so that pod borer cannot eat it. In NIPP people are working. But we saw the wild type Cajanus platycarpus is not susceptible. It is tolerant to pod borer. So we sequenced the genome. Cajanus cajan genome was available, but Cajanus platycarpus genome was not available. Then we have comparative genomics analysis. After that, comparative metabolomics analysis. Then after that, we found several compounds which are either less or more in two different systems. So we procured this compound, isolated some of the compounds, and we went on analyzing. These are the compounds. So the insect were being fed with these compounds. These are anthocyanin, these are other molecules, these are different type of secondary metabolites. And one epicatechin 3 gallate, it was found that this particular compound is giving tolerance to that particular insect. After, I am not talking about computational genomics. I know less about computational genomics. I depend upon other people to give the result. Ajit. So other people, they work for me and they give the result so that we can interpret and go for the wet lab experiments. So this particular molecule, we actually what we did, we found out this particular gene is defective. This particular gene is defective in Cajanus cajan in comparison to platycarpus, as well as the promoter of this gene is having large number of MAB elements for better expression in platycarpus in comparison to Cajanus cajan. So we knew the cause, how epicatechin 3 gallate is less in Cajanus cajan in comparison to Cajanus platycarpus. Started from the genomics and went to the molecules. Then this I told about the MAB elements. Then we transferred to tobacco, and in tobacco we started analyzing how the insect is being inhibited, epicatechin 3 gallate. Then what happens inside the insect? when you feed the insect with epicatechin gullet. Then we got that lipogenesis is being decreased in the insect and lipolysis is being increased. All the genes of lipolysis were increased. So that's the cause why the insect is becoming less weight, smaller, and gradually it will die. Another example, this is in Mentha piperata. This actually viridiflural, and molecule, small molecule called viridiflural. And interestingly, this molecule, if you spray it, is having high fragrance as well as it induces for lactation, better lactation. And these genes were not being analyzed till now. So these molecules is being known. Some people they have got the molecule, isolated the molecule, genes were not being. So we got the molecule and we found out, also pathway analysis, I am not going to that. So it also inhibit the insect, Bihar hairy caterpillar, which infect the mentha plant. So there are specific molecule, and what we did, we overexpressed the mentha plants which produce 0.5%, 0.05 to 0.5% to 25% in the essential oil. When it is higher amount, the insect didn't grow. Earlier the insect were killing that and infecting and killing that. Again, in Artemisia annua, what we did, again RNA madam has told, Subhra madam, she has gone. Then cinnamic acid, this is the phenylpropanoid pathway. If you go for RNA of C4H portion, then what will happen? All these things will be deficient. This will not be produced and cinnamic acid will be accumulated. If cinnamic acid accumulated, then benzoic acid, it will induce to benzoic acid, benzoic acid will be produced, benzoic acid, salicylic acid, and salicylic acid, you know, elicitor. It increases the artemisinin content in the plant. But mind you, lignin is decreased, plant become lanky, xylem and phloems are less formed, and phenylpropanoids are not there. So what happens? This became susceptible to insect, sucking insect and pest. Also many fungi. So after knowing that, we induced artemisinin, higher amount of artemisinin, plant become lanky, but it became the host. This was earlier to host to some insect and pest, and not host to this particular pest. Now it is coming. Also some of the fungi earlier not infecting to this plant, now after reduction of the lignin content or phenylpropanoid content, it became they started attacking this plant. So what we did, we overexpress PAL gene in Artemisia. So when we overexpress, the cinnamic acid will be more, also the lignin pathway will go on, artemisinin content will be more because of the cinnamic acid, as I told earlier. Benzoic acid and salicylic acid, this pathway, it will be elicitor and eliciting. Then when lignin pathway is more, phenylpropanoids are there, the plant earlier susceptible to those particular insect is now not susceptible. The plant susceptible to fungi, different type of fungi, is not susceptible to the fungi. So that's the beauty of secondary metabolites for the internal immunity of the plant. These are the experiments, how they are not infecting, which was infecting earlier. Again, so this is the pathway. This is the pathway of menthol biosynthesis. You see different type of species are there. There are blockage in different parts of the pathway. For that reason, different molecules are being produced. And one of the molecules, menthofuran, and this menthofuran produced by Mentha piperata and not by any other species. And believe me, because of the menthofuran, the plant is not susceptible to Bihar hairy caterpillar, and all other species are susceptible to Bihar hairy caterpillar. So why it is like this? This is because the intron, the genes are there. I told the synteny concept earlier. The genes are there, and it is governed by, there is a single intron inside that gene. Because of the GT, they have GTAG boundary, very nicely textbook type. But when there is a acceptor arm deficiency, there is a mutation in the acceptor arm, there is no formation of lariat, or the intron's length is too small and there is no acceptor arm at all. So they are not being spliced out, or everything is perfect in the intron, but there are mutations in the downstream of the ORF, for that it is not forming the functional protein. So how these appeared? Probably Mentha pipirata was going in a region where there was high infestation. In all other region there is no less infestation of this particular insect. Maybe, who knows, but this is the case. Coming to Tulsi, I have 9 Tulsi variety. Our team, we have developed in CMAP. I will not talk about much more, where there is genome, maybe Madam Sodamini will be speaking. We actually, from India, first time, we both together simultaneously we reported the Osimum genome. So there are 9, we took CMAU. So this is having 26, 16 chromosome there it is having. So I will not talk about this thing, phenylpropanoids and all other gene. And lotus, recently we have a lotus genome. Lotus, this is a 108 petal lotus. It is discovery that this particular lotus contain 108 petals. And how I will just, this is not about the computational genomics, a little bit diversion. I wanted to show you. So this is how it related, the genome is related, and we have complete analysis of all the nutrients, metabolites, everything together. The genome analysis, all the analysis, how the metabolites are there in the chromosome, how the different from other genome analysis, and we have the genomic resources of these plants and complete genes, and some of the molecules which we get feedback from the commercial sources, we go for this gene. So there are some important molecules which are in the fragrance and flower industry. So these many genes, terpene synthases, we have isolated and 21 cloned, and gradually we will be knowing what they, in addition to the fragrance and flower, how actually they are behaving in immunity. And these are the genes. And one of the genes I told about viridiflural, which is this one around 10 milligram $300. This is high viridiflural compound, behave like a hormone, also insecticidal in nature. And see the number of, I was talking about vetiver, the number of molecules. And this particular plant, recently the paper has come out in phytochemistry. This particular plant is in the root, if you analyze, these are the molecules we have analyzed, but there are many more molecules. And believe me, this plant is tolerant to insect pest. This plant is tolerant to all type of stresses like water stress, water logging stress, salt stress, heat stress, cold stress, everything. So there is a faith, trust and belief that in future we can beat the climate resilience, we can have climate resilience plants, in crop plants, by searching the secondary metabolites. And I told you earlier that we have forgotten the secondary metabolites. We are searching medicinal plants for our immunity, and but we are forgetting that already the plants, crop plants earlier, they were not having, means what we did actually, we went on breeding for yield and yield, because we were getting, earlier we were getting food aid from USA, PL 480 or something like that, and we went on breeding to feed the people. Now we are self-sufficient. And in the mean, a breeder is a good breeder who will actually focus on a specific character, yield, if he is searching for yield, and reject all other plants. So we have lost many secondary metabolites on the way. Now we are trying to fortify the plants, but we will get everything in the wild resources, as I told, platycarpus. And if we analyze, compute and say and design the plants for future through computational genomics, that will be a wonderful thing to happen. This is BRA. Thank you very much. Jai Hind, Jai Bharat. Namaskar.
Questions?
Thank you for the talk, sir. I have two questions actually. First one, is there any cost a plant has to pay if we target some upregulation of some secondary metabolites?
Yes, because secondary metabolites are derived from primary metabolites. So from the primary metabolites, the flux, I have not told about all other, several other things. I also talk about the metabolite channeling. So I work on that. I have given a small part of the work. Metabolite channeling, how it happens. So when it is drawing carbon or nitrogen source from other sources, from the primary metabolites, definitely there will be a cost. But the cost is good enough to survive for the plant and get the immunity along with the crop products to you. So you may be having a little bit of yield penalty, but that is worth having in the plant.
Okay, sir. And one more question is, which secondary metabolites would you suggest, which can give broad spectrum tolerance to multiple fungi or other stresses?
That has to be searched upon. And for broad
Broad
spectrum,
spectrum.
like just like fusarium, it is a broad spectrum fungi. So it attack many crop together. Rhizoctenia, Puccinia, what Madam told, Subra Madam told, many which are having highest in the top 4 or 5 categories, that broad spectrum. So you have to search for broad spectrum metabolites and keeping in mind the synteny aspects. Maybe you can, when you compute, analyze all these things, get the databases together, enter into it and find out for me through AI which are the molecules, then I can go for the wet lab analysis.
Thank you, sir.
Sir, Z3 hexanol has been used means are you able to identify resistant line through Z3 hexanol in tomato leaf curl virus for particular?
This is not my work. Okay. This is the work from the literature, but tomato leaf curl virus. First example I gave it from others' work. Rest of the examples of mine. So okay. These first came out. So okay. I don't know whether they have isolated, but hexanol. When have you ever experienced a smell of fast rain? Okay, when you are moving, the raindrops are falling down. That's the smell of hexanol. So the actinomyces, when the raindrop falls, this smell comes out. And these particular small molecules gives work that much. It gives immunity to the plant.
Thank you, sir.
Sir,
Just a question quite different from. May I?
Yep.
Yeah. So it's different from the questions which are coming from the others. I'm curious to know the Nilumbo lotus petal number. Is it a continuous gradient that you see within the various species? There's a variable number. You mentioned that one particular species has only 108 petals.
One specific accession. This is
Accession.
Nelumbo nucifera is the species.
Okay.
So we have got a collection of Nelum nucifera.
Uh-huh.
So from different part of the country, and when they have not been analyzed totally. So when I joined, then I told count the petal. Luckily, we got from one accession 108 petals. So we domesticated that, and earlier it was not flowering for.
And is it stable? So
Stable, yes.
No, that's my question.
Yes.
The question was on stability.
Yes.
Or whether you see any fluctuation or variation in the number of petals.
There is a collection from 7 or 8 years back.
Right.
And it was where from they have collected. We went and analyzed that. That was also 108 petal. And now also 108 petal.
And it's
year
stable.
after stable.
Okay. And its closest succession, what is the number?
There are 147.
Okay.
1000 also.
All right. So it is a reduction in the number. All right. Thank you.
sir, it was an impressive talk. My question is on, so what's your comment on homeopathy, human consumed homeopathy medicine is been well performed under in vivo, in silico, and in vitro against various soil-borne phytopathogens and secondary metabolites of medicinal plants. Were they have high stable in vivo conditions?
See, homeopathy, I don't have much knowledge. I should not comment on that. But still, I like to tell secondary metabolites, they are being used in reductionist approach in homeopathy, what I know, little bit about that. What I say, if you are in less quantity, the effect is very different than if you take in a higher quantity. Beyond that, I have no idea. Homeopathy, how it gives immunity and all this thing.
Yeah, actually, thank you, sir.
Yeah, so here, so the secondary metabolites that you have deciphered for this vetiver, is it just the combination and amounts of the secondary metabolites that are different, or did you find any small molecule which is only found in this plant that gives that unique flavor and so on?
Yes, there are some molecules, betivone,
Yeah.
fusimol, fusinone. These are the molecules which are actually giving the flavor in vetiver. And they are highly defined molecules. These molecules are only not only for taste, they are highly antibacterial, antifungal in nature.
Ask you to discuss or deliberate. See, while secondary metabolites are very expensive to make, expensive in terms of the energy investment that the plant will have to put in order to make it. So if we want to boost the amount of this, cocktail of secondary metabolites, it will be even more expensive. But more importantly, it shouldn't cost on the yield penalty, because if you want to protect either the roots or the shoots without affecting the quality of the agricultural product, whether it is the pod or the seed or the grain, you have to very, very carefully spatiotemporally control it and prevent any yield penalty.
Agreed.
Are there people doing these analyses? Because
Yes.
in theory, it looks good, but plants have evolved to balance their output to the amount of input they have and the kind of nutrients they have. So in theory, it looks very nice, but one has to consider these other points. Yeah.
Yeah, the work is going on, madam. If you, I mean, those are present already in the wild types. Wild types, there is yield is less, but the secondary metabolites are higher. But now question is that when you have higher yield, can you introduce the secondary metabolites with very less penalty or having no penalty? Can you find out some of the factor what is happening? Maybe by inducing more of the carbohydrate production or something like that, photosynthesis. These things are there to correlate.
Can you also track the endophytes that, so those are the secondary metabolites, endophytes in the plant. So are you looking into that kind of microbiome studies where you
Yes.
could enhance?
Simultaneously, I told some of the endophytes or some of the biostimulants are of that nature only. They complementary, they stay in the plants, either they are in the rhizosphere or they are inside the plants, helping the secondary metabolite production, or maybe introduce those things for the secondary, but still then they will be depending upon the plant carbon to produce
Yeah.
more.
Yeah.
Any other questions from the students?
Thank you, sir. Sir, actually, you have said about enhancing the plant secondary metabolites in the plant. But sir, if you want to extract the compounds, then it's fine. But the plant, if it is edible, and as because these secondary metabolites, they give fragrance, or the increasing amount of these metabolites, it might affect our health. So have we considered
Yeah.
those things?
See, nothing to be. One peg you take, two peg you take, three peg it is okay, four peg you take, five peg okay. If you take one, two, three bottle together, then you will die. like the secondary metabolites. Very small amount, not cytotoxic, it is produced inside the soil. Little bit more amount, it will be expelled to a specific structure like trichomes and all other latex vessel, latex tissue. Like this, there is concentration dependent. Very small amount, it will be helpful, but for higher amount, it will be definitely.
So, sir, because if a particular spice, if we increase this particular secondary metabolites, so the customers or the consumer, they have to know, right, that the amount which they are taking it
Okay,
generally.
okay. See,
Sir.
you like a strong spice. Some other person require a very less strong spice. The Indians are spice loving people. So secondary metabolites may be higher in that particular. But if you increase the spice, if secondary metabolites in the spice, or take more spice, पेट खराब होगा ना?
Yes, sir, definitely, sir. Therefore, I'm asking.
हाँ,
Because
पेट
we have
खराब
to
मत
know, no,
करो.
sir.
हाँ?
Sir, we have to know that whether this particular plant has the metabolites are increased, or we so or this transgenic plants are being, or the products are being incorporated in the market. If they come out in the market, sir, we have to know
No,
about this, sir.
transgenic, we don't do that actually. Good. Then
Yes, sir.
Change and all these consumer questions come in. So that is related to the question that I was asking Dr. Ajit to discuss. Yes, we can put the secondary metabolites, but if you put them all over the plant, it will be there in the fruit. It will completely alter the
Tastes.
characteristics that the consumer is looking for. So definitely, you have to control where and when you make these compounds.
I'm telling
Everything
about
is in the right time and place. It is very good.
a small amount of metabolite which will bring back the immunity of the plant of rice, wheat, maybe pulses. So I'm talking about a small amount of metabolites. For a higher amount of metabolites for spice purpose, if you eat it, I gave you example. If everything excess is bad.
Yeah, I think there are a lot of questions still. We are running short of time. So thank you, sir, for that enlightening presentation.